Hello everyone. Welcome to the Food Tech Networks. This is Astha Sharma, and today we are going to discuss food microbiology MCQs part 2. And before starting, if you have not subscribed the channel, do subscribe. And for all the latest notifications, please press the bell icon. So let's get started. The question number 1: who developed the method of preserving food using boiling water? So the options are A. Peter Durand, B. Louis Pasteur, C. Francois Nicolas Appert, and the last option, option D, Charles Michel. And the right answer is option C, Nicolas Appert. So what happened over here is this Nicolas Appert introduced method to preserve food using the heat from boiling water. And Peter Durand is credited with the development of the steel canning method. And Charles Michel, he incorporated the metal lining inside the cans to improve the quality and efficiency of food preservation. And of course, the pastor introduced heating of wine to destroy soaring bacteria. So now the next question is question number 2, which is what is a full form IMF? And the options are option A, inner moisture foods, option B, intermediate moisture foods, option C, inconspicuous moisture foods, and option D, inane moisture food. And the right answer is option B, that is intermediate moisture foods. So intermediate moisture foods are those that have water activity between 0.85 and 0.6. So this inhibits the growth of gram-negative bacteria. More the water activity, more will be the microbial growth, because free water will allow microorganisms to proliferate. Question number 3: what is the primary purpose of adding chemical preservatives to food? And the options are option A, improving texture, option B, enhancing flavor, option C, increasing nutritional value, and option D, that is inhibiting microbial growth. So the answer is very sure, inhibiting microbial growth. It's very no, usual thing. So the explanation is chemical preservatives are added to food to prevent or slow down the growth of microorganisms, thereby increasing the shelf life of the product. So microbial growth is directly related to the spoilage, which will obviously it will lead to economical loss of product. And of course, if we are inhibiting the microbes, then if we are we are successful in inhibiting the microbes to grow in our food, then obviously the shelf life of that product will be increased. So the question number 4, which is in the context of food microbiology, what does HACCP stands for? Option A, hazard analysis and critical control points. Option B, high altitude cooking and cooling process. Option C, healthy and certified culinary practices. And option D, highly advanced cold chain preservation. And of course the answer is option A, that is hazard analysis and critical control point, option A. So it is a systematic approach to identify, evaluate, and control the food safety hazards through the production process. In If we talk about the chart of this HACCP flow chart, it actually contains 8 points. So it's very important to go on with HACCP in each and every food industry, so that you can minimize the risk while production of any food item. So HACCP should always be considered as your first choice, or first step to go through while you are producing anything while production. So the question number 5, the name Staphylococcus has dash origins, and the options are A, Greek, B, French, C, Danish, and D, polish. And the right answer to this question is option A, that is the Greek. uh, Staphylococcus has Greek origins. So what happens over here is there was a Scottish surgeon who first described Staphylococcus as the cause of pus-forming infection in humans. So he coined this name in 1882 after their appearance under the microscope. In Greek, Staphyli is a bunch of grapes, whereas coccus is a grain or berry. And if you focus means obviously green or berry, and if you see this staphylococcus under a microscope, so these are in clusters and are coccus, or we can say it's circular in shape, and these are in clusters, staff, and it's like in chains or bunches, we can say. So this staphylococcus aureus is a thermophile, mesophile, saccharophile, or thermoacidophile. So the correct answer to this is option B, mesophile. Staphylococcus aureus is mesophile. Uh, and for mesophiles, the temperature range for these mesophilic organisms is that they grow at a moderate temperature, and hence and therefore their name is mesophile, and it operates under a temperature range of 280 to 321 degrees Celsius. And we all know that Staphylococcus are is a very common microorganism which is every time we can say it's present in on our skin and on our hands, on our body. So and for its growth it requires normal temperature or body temperature, we can say. Thermophiles, they require higher temperature. Fine. So Staphylococcus aureus, it requires medium type temperature, moderate temperature, we can say. Staphylococcus aureus enterotoxins are super antigens. The question number 7 is this. So, and it is like true or false question. So the answer to this question is yes. Option A, true. So these toxins are able to get the body to produce a large percentage of T cells than any other conventional organism. Hence, it is the reason that's why Staphylococcus enterotoxins are superantigens. And what's the difference between antigens and superantigens? It's that if we talk about antigens, these conventional foreign antigens, we can say these are recognized by T cells, right? Within the MHC uh, uh, and of MHC, peptide binding group is there, and these are recognized by that. So super antigens, different from these conventional antigens, they bind with high affinity to class 2 MHC. Okay, whereas normal antigens only MHC, and super antigens class 2 MHC molecules outside the antigen binding group in the absence of antigen processing. So this is what the difference is. Antigens usually bind with this peptide binding group of T cells within the MHC, and these super antigens bind with class 2 MHC outside the antigen binding group in the absence of antigen processing. So the question number, next question, question 8. Staphylococcus Staphylococcus aureus toxins are myotoxins, necrotoxins, cyto uh, myotoxins, uh, uh, cytotoxins, and option D, neurotoxins. So the answer to this question is option D, neurotoxins. Why these neurotoxins? So what happens? This, I mean, this neurotoxin, this toxin works on the gut receptors actually, and it sends signals to the vomiting center of the brain through nerves. Hence, these are the neurotoxins because they are affecting the nerves, or we can say the receptors of our brain. So there are other, in short, just for information, there are other toxins also like alpha toxin, ochratoxin, fumonisins, So there are different organisms which release these toxins. For example, if we talk about alpha toxin, Aspergillus flavus is related to that. Aspergillus parasiticus, Aspergillus alpha toxiformans is there. Ochratoxins also Aspergillus की ही subspecies रह जाती हैं. Ochratoxin they can release. Fumonicins वो भी Aspergillus और Fusarium दोनों की species जो है release करती है. So the question number 9, which is coagulase can cause a coagulation of blood plasma even without option A, potassium, option B, iron, option C, calcium, and last option D, magnesium. So the correct answer to this question is option C, that is calcium. Coagulase is an extracellular substance, okay, which can be produced by Staphylococcus aureus. Its presence can be detected by EDTA kits. Ab EDTA kits, these are, is already market में available रहता है ये. हमें कुछ बनाने की जरूरत नहीं होती है. These are EDTA tubes already company made आता है. So these and we can use this EDTA test to assess the function of whatever we are dealing with, whether our kidneys or whatever, through a series of blood tests. So EDTA kits are over there. So what is the full form of EDTA? It is a ethylene diamine tetraacetic acid, that is EDTA. And what is it actually? EDTA is a polyprotic acid. It contains 4 carboxylic acid groups and 2 amine groups with lone pair electrons that chelate calcium and several other metals. So why this EDTA is used? It is it is used to treat acute and chronic poisoning also in our from blood stream. In this EDTA is used as medicine also in that things. So the next question is question 10. What genus of bacteria causes botulism? And the options to these questions are the option A, Escherichia, option B, Salmonella, option C, Vibrio, and option D, Clostridium. And the correct answer to this question is option D, Clostridium botulinum. So this Clostridium botulinum is a rod-shaped bacteria, anaerobic. It has the ability to form spores, and it's a neurotoxin botulinum. So neurotoxin again, it is related with the brain's receptors. Clostridium botulinum, and it is very much present. Very much I cannot say, I should not say that, but it is linked with the canned food poisoning. If canning, if the canning has not been done properly, if there is any bacteria, then botulinum can be happen with this canned foods. So now botulism is named after option A, ham, option B, sausages, option C, sorry, beef, and option D, mutton. So the correct answer to this question is the option B, sausages. And the explanation to this question is botulinum is named after the Latin word for sausage, that is a botulus. This is because the first reliable case of botulism was caused by spoiled sausages in Germany. That's why its name came from these sausages. Question 12. Difficulty in swallowing is also called dysphonia. Option A is dysphonia. Option B, dysphagia. Option C, dyspnea. And option D, dystonia. And the right answer to this question is option B, that is a dysphagia. And the explanation is botulinum toxin causes difficulty in swallowing by attacking the muscles, controlling the process. This is a side effect of Botox as well. For extra information, I must tell you that this cholesterol is Clostridium botulism does not cause only botulinum disease. It is used in this, this this company in which we can, pharma, if we can say whatever. It's Botox, which usually people do for their face so that their skin should be more tighter and more apparent and more glowy or something like that. So this Botox came from this organism also only, Clostridium botulinum. Botox is produced from this organism. Next question, question 30. Botulism toxin causes flaccid paralysis. And this is again a true or false question. And again, two options, option A true, and the option B false. So the correct answer is option A, true. The botulism toxin travels up to the bloodstream through the lymphatics. They bind to the nerve ending of a muscle nerve junction. This blocks the production of acetylcholine, which transmits timely. Hence, it gives rise to flaccid paralysis. A flaccid paralysis क्या होता है? It is actually a neurological condition है. We can say, and it is characterized by weakness or paralysis and reduced muscle tone without other obvious cause. Example, trauma नहीं है, कुछ नहीं है. This is all. These are the only symptoms, and you know. So जो nerves involved रहती है muscles के साथ, ये उन पे act करता है actually. That's why flaccid paralysis, botulism ये cause कर सकता है यहाँ. So now question number 40. Mycotoxins are produced by option A bacteria, option B viruses, option C protozoa, and option D fungi. And the correct answer to this question is option D fungi. Explanation is some molds produce enzymes and toxins called mycotoxins using food as a substrate. These chemicals can cause a wide variety of diseases ranging from food poisoning to cancer. So mycotoxins are produced by fungi. So this is all about this question. Now the question number 15. Alpha toxins are produced by option A penicillium, option B aspergillus, option C Saccharomyces, and option D Rhizobium. And the correct answer to this question is option B aspergillus. Aspergillus flavus produces these toxins which are known as aflatoxins. Then these are highly carcinogenic in case of rats, and of course in case of humans also, but mostly in case of rats, the studies has been done on those. So they exhibit fluorescence under UV light also. So this is all about aflatoxins. These are produced by mostly two species of this aspergillus. They're actually a aspergillus. So now the aspergillus flavus, and the other is the aspergillus parasiticus also. Now the next question, question 16. Aflatoxin B1 epoxide reacts with dashed residues of DNA. The options are option A guanine, option B adenine, option C cytosine, and option D thymine. And the correct answer to this question is option A, that is guanine. The toxin acts on the P53 gene. This gene has an anti-cancer function. Also, the hepatitis B virus binds to the product of the same gene. So hence this could prove to be either a bone or a bean. So this aflatoxin B1, it reacts with the guanine residues. Fine, and this P53 gene is a very hot spot for researchers for cancer treatment or cancer or drug designing. P53 gene. So the question number 17, which is ergotism, is caused due to the and the options are the Option A, Aspergillus niger. Option B, Penicillium notatum. And option C, Saccharomyces cerevisiae. So, and option D, Claviceps purpura. And the right answer to this question is option D. That is the Claviceps purpura. And this function grows on rye and other cereal crops. And the alkaloid ergotime causes hallucinations, nausea, uterine contractions, etc. So, what exactly ergotism is? Actually, it's a long-term ergot poisoning, and traditionally due to the ingestion of alkaloids produced by the Claviceps purpura. And as I told you, this infects rye and other cereals, and more recently by the and it is also known as the ergotosis, ergot poisoning, and Saint Anthony's fire. You should remember this. Saint Anthony's fire is related with ergotism, and it is caused by the Claviceps purpura. And symptoms to this are, this nausea and uterine convulsions. All these are under, these symptoms. And this is all about this. Edema is also there because of this. And loss of peripheral sensation is also one of the symptoms. Vasocontraction, as I told you, uterine contractions. So gangrene is also one of these symptoms. So this is all about ergotism. Now the question number 18. Ergotism is also called option A, San Xavier fire. B, option B, San Anthony's fire. B, option C, St. Paul's fire. And option D, St. Matthew's fire. And the correct answer is, as I told, St. Anthony's fire. Ergotism was called St. Anthony's fire as the monks of the order of St. Anthony were successful in curing this disease when it was rampant in the Middle Ages. So that's why it is called as a St. Anthony's fire. Next question number 19, which is a minimum, optimum, and maximum temperatures of an organism can grow at or called dash temperatures. And the options are option A, cardinal, option B, major, option C, key, and option D, critical. And the correct answer to this question is option A, cardinal temperatures. In the graph of growth rates versus temperature, there are three cardinal temperatures represented by 3 points. That is minimum temperature, optimum temperature, and maximum temperature. These tell us about the characteristics of a particular organism and are different for different microbes. The classification of microbes can be based on their cardinal temperatures. So, last question of question number 20. Dash can be frequently found in humans and animals because of their optimal temperature. And the options are A, psychrophiles, option B, mesophiles, option C, thermophiles, and option C, psycherotrophs. And the correct answer to this question is option B, mesophiles. And we, as I told you earlier in early question also, mesophiles have an optimum temperature of around 37 degrees Celsius, which is our normal body temperature, which is average body temperature of humans and animals. Mesophiles thrive quickly and hence contribute to faster spoilage or spoiling of food. Some mesophiles are, for example, Salmonella and Clostridium perfringens. So, thank you. That's all from my side. We will meet in next video. Till then, take care of yourself and so do subscribe our channel. Thank you.